we treated these mice only with a single injection of this microRNA, or we saw that these mice stopped degenerating, they stopped deteriorating, and behave almost like normal mice, or like normal mice in most of the properties.
When a treatment is effective, you don't really need statistics to prove it.
People living with ALS always ask the same question: how long?
In 5 years, I'm really hoping that we'll be in a phase 3 or even using this drug to stopping or treating, curing ALS.
I'm Tim Green, and I have ALS. But this podcast isn't about ALS. I don't want you to feel sorry for me. I'm a father of 5, married for over 35 years, a New York Times best-selling author, an NFL first-round pick, and a practicing attorney. This is Nothing Left Unsaid, where my son Troy and I sit down with extraordinary people for raw, honest conversations about life, its toughest trials, and greatest triumphs. My voice in today's episode is powered by 11 Labs. Before we go any further, can you bring our listeners up to speed? In plain terms, what did you discover and what made you realize this was something fundamentally new in the ALS story?
So actually, our story starts back in around 2021, when we realized that a protein that probably many of ALS patients already heard about, TDP-43, is found not only in the cell bodies of ALS patients, where it normally is being known to be aggregated. So we actually found that this protein is found also in the peripheral ends of the nerves, right next to the muscles, where it tends to aggregate and affect the stability of these synapses that link between the nerve and the muscles. And our current or more recent findings suggest a mechanism that can control this protein, this TDP-43, and maybe regulate its accumulation at the synapse, preventing the degeneration of these synapses. Amir, if you care to add on your hand.
Yeah, so actually, this started as a friendship collaboration because Ariel's mentor, Eran Persson, the head of the lab, was telling me about their system in the lab, and that they were examining the accumulation of this TDP at the lab. And I was telling him, I do muscle biopsies, and I see these nerves in muscle biopsies. Maybe I can look there. And sometimes it's muscle biopsies that show ALS. So we started looking in these muscle biopsies of patients, and we found that the TDP aggregates in the nerves in the muscle biopsies. So that's sort of straightened the idea that the TDP has a lot of effects also near the neuromuscular junction, which will come back later. Anyways, that was the first thing that was really striking, and we published that, as Ariel was saying, in 2021. And we found out that these TDP aggregates, they sort of capture RNA, which are important for producing proteins, and proteins, are essential for energy, for production energy. And if that's not functioning, then you lose your energy and the nerves degenerate. So that was a few years ago. The new thing that we found was that there's very low amount of this special molecule, this very small RNA called microRNA, specifically one that's called 126. And we found that Ariel and the team found that it's very low in animals that have ALS. And we were really surprised about this. So we went back to the patients, and I asked some of the patients in my clinic to give some samples of blood to see if that's low in their blood. And we were surprised to see that actually that is very low in the blood of patients with ALS. So it sort of made sense suddenly. And the last thing in this story is that when we injected this small molecule, the microRNA 126, into the blood of animals that are sick with ALS, that really affected the disease. It stopped the severe progression of this disease, and it even grows the nerves back. It really helps in regeneration of these nerves. So all of that together was sort of striking because first we found the mechanism, or at least this part of the mechanism of this disease. And second, we found a molecule that can affect this mechanism, and actually as far as we can see, it stops the disease progression and even helps in regeneration of some of the nerves. So we're really hopeful that this is a meaningful, accomplishment, and that this will be, open treatment opportunities using this molecule. Yeah.
Most ALS research is focused on the motor neuron as the primary point of failure. Your work shifts attention to the conversation between muscle and nerve. What were we missing by not looking there sooner?
If we go a few years backwards, we didn't know that these nerves, or the junctions between the nerves and the muscles, function as almost an independent unit, independent cell. Almost all cellular processes also take place there in the nerves. And that shifted the focus to look also at that end of the motor neuron. And our recent finding, as Amir just told you, that identified that TDP-43 also accumulates in these nerve endings at the neuromuscular junctions. And this happens as far as we saw very early in the disease, in the clinical stages of the disease. We thought that this is very important and very crucial. And if we can intervene in this aggregation of TDP-43 at these early stages, before the synapses degenerate, before the nerves degenerate, maybe we can also stop the degeneration of the motor neurons themselves.
I know some of these studies are in my animal models and some are in people. Do you know which one's first? In the TDP-43 abnormalities in the axon near the nerve muscle or in the cell body? Like, can you tell which comes first, at least from the animal or cell work that you do?
Well, when we looked in animals, first of all, because this is the big mass of data we have is from animal models, so we see that these TDP-43 starts to accumulate and you don't see any impact on the synapse. So it accumulates locally at the neuromuscular junctions before you see any pathology in the spinal cords, in the motor neuron cell bodies of the spinal cord. Our recent article shows that these TDP-43 are formed locally through a process that's called local protein synthesis. So if we talked before about RNA that serves as a template to create proteins, so one of the RNAs that we found at the synapses, at these neuromuscular junctions, is the RNA of TDP-43. And for some reason, or now we know the reason, because the regulator that we just named, microRNA 126, is not there to regulate the production of TDP-43, and then basically it unleashes an unregulated production of TDP-43 at the synapse.
Just to add about that, when I first started these discussions with Ann Perlson years ago, and he told me, this disease starts at the neuromuscular junction. I was surprised, and I told him, no, you're confused. It's a motor neuron disease. It starts in the anterior horn of the spinal cord. I mean, that was what I was taught. That's what we relate when we do EMG, this test to identify the motor neuron degeneration. But the fact is, as Ariel was saying, the accumulation that we see is really in the neuromuscular junction and along the nerve that we see in the muscle biopsies, which is very, very close. This is, a centimeter away from a neuromuscular junction. So at least from our point of view, this is where this disease starts.
So when somebody without a medical degree hears it, why wouldn't we be like, if it's working, right, and we're taking this specific RNA, I think it's like 126, and we're giving it to animals, it's working, and I think we started in people too as well, right? Or not yet?
We didn't test it in humans, but in patients, in human patients, then we found that it's very low in something called exosomes or vesicles that are excreted from other cells. The largest organ in the body is the muscles, so probably lots of it comes from muscle. So in other words, we found that it's very low in these vesicles in humans with ALS. So likely it's also low in the muscles of these patients. That's something that we're testing right now, actually. And it makes sense that if we treat that in humans, we'll get the same effect. That's what we're hoping. Furthermore, Ariel can tell you about the human cells that we tested in the lab. I mean, everything was done both in animals and in human cells tested.
So what's the jump from animals to people? Like how long will that take, and what's the process there?
I think Merit is the most experienced one. I can tell from my point of view. I should have added the disclosure that nowadays I'm working at a pharmaceutical company in Israel called Teva Pharmaceuticals, but whatever I'm saying here is based on my opinions. So the process is not that simple. You have to go through various processes of verifying the safety of treatment, developing the delivery systems. How can you deliver this small microRNA specifically to the target, to target motor neurons, or specifically to target the nerves, and to validate basically the entire mechanism that we identified in human cells, in human models. So it's not that fast, but Merit can expand.
Well, hopefully it can move fast, but especially in a field like this where there's such a need to be fast and so many people interested in it. I'll say a couple things. One is your work is fantastic, so congratulations, and it's great. It's really exciting to hear for a number of reasons. One, obviously, the therapeutic potential. So there it is, exactly what Ariel said. You have to figure out how to deliver it, and whether that's going to be through a gene therapy approach or another approach, and get to the right target. So that's one step of it. And then the other is actually figuring out, is this something for everybody with ALS? And it might be, but that needs additional human studies, like what Amir was talking about, looking at maybe muscle or blood from a lot of people with ALS to see if it's present. Can you detect this change in everybody? And that'll be enormously helpful for the trial. So you could do this work in parallel, kind of develop the drug delivery approach and really do the studies in people to see who you want to target this treatment for.
Would the blood samples that we already have for all the ALS patients all over the world, could we use those to check, or it has to be new samples?
We have plenty of blood samples from patients, but the idea, and we think that the same mechanism that we saw in animals is present in humans, because we found the deficiency, the reduced levels in human blood, and Ariel used human samples to test all the mechanism. Next thing to do is we need to inject this molecule to humans, but before we can do that, we have to find the right way to deliver it. We have to make sure that it's not toxic, doesn't do any harm to humans. We have to find the right dose. There's a bunch of different things to do. Sure. And that needs time and lots of money.
But Amir, when you guys were looking in ALS patients, were you looking at patients with both, genetic ALS and sporadic ALS, and everybody was deficient regardless?
Actually, all the patients that I looked at were all sporadic.
Okay.
But we think that the same mechanism is happening in both genetic and sporadic ALS. We think that's the same thing. Actually, many of the, or actually all of the laboratory studies that Ariel did was on genetic ALS, because it's cells that were grown or taken from people with genetic disease and sort of used in the lab.
And then the last question on this topic, and I swear we can move on, but in the United States, we've got the FDA, which I think ALS patients would agree merits. This is not Merit saying it. This is me as a patient family member. The FDA is so slow with stuff like this. Does Israel have that same problem, or can you move at faster speeds than you could if you were a U.S. based research team?
It's different. We have Ministry of Health regulation, okay, and things can be done in Israel maybe faster. However, we're a small country, and eventually things will run forward faster if it's done, maybe most of it outside of Israel and regulated, accepted by the FDA and the European regulation and that sort of stuff. So we'll use both.
You kind of hinted at this already, but was there a moment in the data when you thought this doesn't fit the model we've been taught, a moment where the old assumptions started to break down?
Yeah, so from my point of view, there were several of these points along the way. The first one, as we just described, was the identification of these TDP-43 in the nerves, in the neuromuscular junctions. But the second one came from finding it in a very unexpected model of ALS. Perhaps the first familial form of ALS was discovered in a mutation in a gene called superoxidase mutase 1, or SOD1, which for years has been negative for TDP-43 pathology. Clinical studies showing or examining the TDP-43 pathology in these patients were negative. They didn't show any aggregates of this protein. However, nobody really looked what happens at the synapses. And we had this crazy idea to look at peripheral nerves. We came in contact with some researchers from Italy that actually published a work that validated our findings back in 2021 and showed on a larger cohorts of patients, they showed the same thing, that TDP-43 is found and accumulates in the nerves of patients at early stages of the disease. And we asked them, did they ever see it in SOD1 patients? And the response was very quick and positive that they did see it before. And this really pushed us to look in the mouse models of TDP4, in the mouse models of SOD1, sorry. And it was striking for us and really got us thinking of how this TDP-43 gets into the neuromuscular junctions. And it drove basically all the research forward on our hand.
That was a fundamental change, because this mouse model of SOD1 is key to testing the molecules, okay, and the therapeutic, intervention. And we were lacking that because we thought this would not affect this model. But eventually we used this model and injected treated the animals with this microRNA 126, Mir126, and then we saw the effect of this molecule, and it was really, really impressive.
I will also add one more interesting point. Actually, in 2018, we already tried this microRNA treatment in these animals and saw the same beneficial effect. Only at that time, we couldn't explain what we see and couldn't link it to TDP43. The change came when we saw the decrease in the patient levels of this microRNA, and when we saw that SOD1, the SOD1 patients also have this TDP43 aggregate. So then we linked everything together and we started studying the mechanism of this accumulation of TDP43.
Whenever someone makes a great discovery, that they're asked then, okay, why did that happen? So my question to you is, do you have thoughts on why the microRNA 126 is low in people with ALS?
MicroRNAs has been described before in ALS, and also actually TDP43 is known to regulate the part of the biogenesis process of these microRNAs. It's still a good question why this happens, why does the levels drop. It could be due to less synthesis, or it could be due to there are these moieties in these cells that basically known to have kind of a sponge effect. They basically take all the microRNAs and sequester them, or it's kind of a sink effect. They take all the microRNAs and silence them as part of also a regulation process on microRNAs. So this could be an option. So actually the microRNA is being silenced by a different, more upstream unknown participant.
I think that we found these beautiful pictures in the paper showing where the microRNA is in the muscle, and it's actually right around the neuromuscular junction. And there's probably a physiological role where it keeps the regeneration, the ongoing regeneration that's needed to keep the neuromuscular junction, modeled as it should be. Because as muscle changes all the time. You run, then you get some breakdown of a few fibers, and you need to remodel the neuromuscular junction. So probably there's this communication between this microRNA and the regeneration of the nerves. However, in normal physiology, you get some injury, you get the regeneration, and that's it. But in ALS, it's ongoing. It's constantly you're getting wasting of the muscle. And I think that's the cause where this progressive drop in this near 126 levels. I think that's the reason. And you can't get over it because you constantly lose more muscle. And this is the intervention that we can offer.
Okay, that makes sense.
I'm going to add a small thing on top of this. So an important thing to know about microRNAs in general is that they don't target a single gene or a single RNA. They regulate an entire group or entire family of RNAs that are part of a larger process going on. It's true also for microRNA 126. It regulates TDP43, but it regulates also a group of proteins called semaphorins and the receptors neuropilin and several other proteins. All together, it seems that they orchestrate the process of synapse destabilization. And as Amir said, we think that this is a dynamic process throughout the life of a normal human being. The synapse degenerate and regenerate, degenerate and regenerate whenever the muscle needs to recover. But for some reason in ALS, this cycle stops and keeps the levels of this microRNA low, creating an accumulation of all these proteins that promote degeneration of the synapse and don't allow the regeneration to happen.
For people listening without a science background, what role does RNA play in this process? And why is it so surprising that it might influence whether neurons deteriorate or recover?
As we said before, so RNA are basically the template to create proteins. We have the DNA that is the genetic code that contains the data that transcribes all proteins in our body. Then we have the RNA that is really the instructions of how to build a protein. And then you have the proteins at the end. And the microRNA that we found is a way to interfere or regulate, better say, the creation of the proteins. You don't want to create all proteins forever. You need to stop this process at some point. And these microRNAs are kind of regulators rather than complete silencers of the protein synthesis of a specific gene or a group of genes. Um, it is important to note, I'm not sure this is the right point, but there are many options to silence genes in research and also in clinical trials, different ways to silence genes. We know that TDP-43 can't be silenced completely. It is an important and fundamental protein that has functions in the nucleus. It also has functions in the nerve, normal functions in the nerves. Only its overaccumulation drives the pathology, it seems. So we don't want to erase it completely. We just want to regulate its levels. And the microRNA that we discovered does it naturally. And we want to control or to increase the levels of this microRNA to bring back the control over TDP-43.
Just for lay term, I like to use the analogy of money. So, RNA is like having a lot of money where you can use it and make stuff, buy stuff, do things. So we need this RNA to make proteins, which the important proteins that we found are missing are proteins that make energy in something called mitochondria. That's the energy source in cells. So if you have RNA, you can use that, you can make proteins, and you can build your energy source. If you don't have it, then you're stuck without energy. It's the same like money. In TDP accumulation just locks all the RNA and you can't use it. It's stuck in the TDP and you just can't use it to make proteins. Just like having lots of money, but it's all locked in the bank and you cannot release it. And you're stuck, you don't have anything to eat because you can't spend the money even if you have lots of it. So that's sort of a simplified analogy.
I like it. We need more of those, Amir. Keep those coming.
ALS has long been described as relentlessly degenerative. Your findings suggest that degeneration may not be the whole story. What evidence convinced you that you weren't just slowing damage, but potentially enabling repair?
So I'm going to start.
Go ahead. You had these observations in the lab, yeah.
Yeah, so we're going back first of all to this vicious cycle that starts by the lowering down of the levels of microRNA. So if it should eventually raise back up, then the regeneration can occur again. In the lab, when we overexpress this microRNA in muscle cells, we saw that the nerves stopped to regenerate and actually they regrow towards the muscles. So there was some kind of, back then we thought it's a repulsive signal, but it's probably, as I said, an orchestrated process of multiple proteins taking part of that allow the axons to stop degenerating and regrow. Basically, you unleash the processes that are blocked there. As Amir told you before, you open the wallets that have been locked, and then you can start spending the money to produce the proteins you need to regrow the axons. But obviously, this comes with a grain of salt when you think of human patients, because there is a limit to a time point that you can intervene. And I think Amir can maybe elaborate on that, or Merit. Obviously, intervening earlier is better, but
I see it like branches of a tree. It's if you cut only the end of the branches, the top branches, they split and regrow. And we see that very well in muscle and nerve biopsies. We see the regrowing, the splitting and sprouting of the nerves. However, just like with trees, if you continuously, chop down lower and the stem branches, then so that's the problem. So it is a matter of time. I mean, definitely, if this treatment works well, and it does help regenerate nerves, the earlier you treat, the better. That's definitely something to note. But going back to the observations, it was striking to see in the lab that if you injure the nerves, which are sick with, these are nerves with ALS model, I mean, with TDP pathology. If you treat these nerves with the Mir 126, you see them growing much faster and much better. So not only that this treatment, as far as we found, it stops the accumulation of TDP in the nerves and the neuromuscular junction, it also promotes the regeneration. So this is great, two things in the same time, which are beneficial effects. So hopefully that'll work.
I'll just add that I think it makes sense. The earlier is always better, but I think we're learning now from some of the gene therapy trials in people with SOD1 and another one, FUS, that we're seeing recovery of function even in people later in the illness. So I think we just don't know. We don't know, in people who've had the illness for a while, maybe the cell body's still okay, and maybe the nerve isn't connected to the muscle, but we don't know that you can't repair that. I'd say that's where we'll in the clinical trials really have to really carefully look at recovery, a function in different limbs and in different patients, and explore it in different stages of the illness. I mean, you could do some of that, I imagine, in the animal models that, intervene at different stages of the illness to tease that out a little bit.
Yeah. So you're actually right. So actually, the nerves have this intrinsic ability to regenerate. Motor neurons, I think, even especially, and sometimes during the disease, you see this kind of event that's called polynervation, right? So when the synapse starts maybe to generate, you see the nerve growing, or either a different nerve or the same nerve grows another extension to connect back to the synapse. So this ability is there, and there's a high the high ability to regenerate, and perhaps this is why you also see the regeneration or the improvement in function in patients with the gene therapy. The muscles are re-innervated by either other nerves or other nerve endings.
In your animal models, what did improvement actually look like? What changed in the nervous system that made you stop and say, this matters?
When we looked at the animal models, these SOD1, the SOD1 animal models, they have a very severe phenotype. They become, or very severe motor behavior. They become almost completely paralyzed by the age of 4 months or so. And when we treated these mice only with a single injection of this microRNA, or with a virus that promotes the expression of this microRNA, we saw that these mice stopped degenerating, they stopped deteriorating, and behave almost like normal mice, or like normal mice in most of the properties. We had some really nice movies showing their gait, the way they walk, and we could really see the difference between an ALS or the ones that didn't get the treatment. They were unable to pass from one side to another of the distance that we were testing in, while the mice that were treated with the microRNA 126 just crossed it without any effort. And this was really amazing to see. And obviously, this repeated itself in many different motor tests that we performed. One thing that I want to add about it is that this also, again, persisted until a very late stage of the mice. So again, we started with one treatment at one time point, and it persisted almost to the age of that these mice normally die or become completely paralyzed. The other interesting point was that when we checked for the expression of the microRNA, we noticed that we only shifted it a bit higher than regular, but we couldn't even get it back to the normal levels of healthy mice, suggesting that this could have even a greater impact once we deliver, and we talked about delivery before, once we are able to overcome some delivery issues and bring this to all the muscles in the body. This will probably help the mice recover their degenerated axons or degenerated nerves and their motor abilities.
Yeah, we tend to say that when a treatment is effective, you don't really need statistics to prove it. You know, the mouse starts dancing, and patients joke around with their doctors and that sort of thing. So the mice didn't dance, but looking at their gait analysis, or the speed of their walking, so the treated animals walked almost as normally as the non-sick mice, whereas the sick mice were far behind. So this was really, really impressive and convincing, even without statistics. We had lots of statistics, but it was obvious by eye.
Did you say that you only had to treat the mice? You treat the mice just one time, and they had that effect? For humans, obviously, it's different than an animal model. Would you guess? I know we don't know yet. But would you guess this is like a constant, ongoing therapy, or is it a one-time thing?
No, so the thing is that when we treated mice, it was with a sort of a gene therapy. We injected viruses that increased the levels of this microRNA continuously. We tried both with different types of viruses, and it worked in all kinds of viruses. Again, the challenge is to bring it into humans. There have been some therapies or there are therapies that use viruses in humans, such as in spinal muscular atrophy, SMA, and they show great advantages for the people that are being treated with. So we think that we can overcome this challenge in humans, and by giving this treatment even once, it would increase the levels of microRNAs for a long period.
I must say, I love all the words we're using: repair and regeneration and return to function. It's exciting to hear those words in the ALS field. That's what season 2 of Tackle ALS is about. And as you were speaking, I was thinking that the Healy Center gives away every year an award for the best team working on repair and regeneration, and they give a talk at the International MND meeting. So I hope your team will apply because that's really what you're doing. So it's exciting.
Thank you.
Yeah, we're excited about this. At the lab, Ariel and Eran are at the lab, but I see the patients and the families. And actually, one of my recent patients is a doctor, is a family physician. And his daughter is a nurse, which works at our medical center. And definitely, this is really important. We're trying to push this as hard as we can.
Scientists are trained to be cautious. What were you most skeptical of in your own results, and how did you pressure test them before believing what you were seeing?
So there are several points in the research where we became skeptical. The first and the most obvious one is that we identified these TDP-43 accumulations or aggregates in a place that no one ever considered them to exist in the SOD1 patients or SOD1 mouse models. And for that, we really had to work hard to verify that our results were real. We used not just one or two, we used several of these mouse models with different mutations in the SOD1. And we started seeing that the results are repeating themselves, and it's not just some hallucinations that we are experiencing. And we continued to work with human cultures of the same mutation, and we also saw that there we see TDP-43 accumulating in the nerve. So all of this together, like the fact that we repeat our experiments and repeat our results, and also in the hands of other researchers, it also repeats, really strengthens our our thoughts about the mechanism that we identified. That's from my end.
Yeah, we test several different, for example, with the biopsies. It's not one or two biopsies, it's several, multiple biopsies. We have disease controls, what we call this, people with other disorders, which are, abnormal muscles, but not with ALS. So we tested that too to compare the situation there, not only comparing to healthy people. So when you see that it's persistent, and after that, after our first publication, we saw similar results by other groups. So that was really reinforcing our thoughts and our findings. That was reassuring.
People living with ALS always ask the same question: how long? Based on where this research is today, what are the realistic steps and timelines between this discovery and a human trial?
Honestly, I think if money wasn't a limitation, right? If you had the money or a company, you could move this really pretty fast to develop the way to deliver it. They're just the tools to deliver, microRNAs using gene therapy, at least for a proof of concept, could be pretty quick. And I would say a year or two maybe would be the fastest, because you have to do long-term safety studies, but it's all about having the resources. And as I mentioned before, I do think we need to do some parallel studies in people, whether it's muscle biopsies or other approaches to really strengthen the work you've already done, to see if it's a treatment for everybody or specific people. But there, I'll go on record that if you have the resources, I think you could do it in a year or two. Most researchers and companies, they spend years trying to raise the money. That's the frustrating part. Yeah. So what do you guys think?
I'm not holding
Sorry.
not holding you to it, but what's the amount of money for something like this? Like just for I know you don't really know, but if you had to guess, are we talking about $1 million, $10 million, $100 million, like for people who don't know?
What do you guys think?
The quotes we have are in the range of $6 to $10 million to reach first in human. Okay, what we call the phase 1, okay, first time in human studies. So passing a variety of different assays in animals with exactly the drug that we will use in humans. Okay, it's a series of experiments to see that it's not toxic and things like that, the distribution of the drug, effectiveness. So all of that all together, including the manufacturing of this virus, these are really expensive stuff. So getting all that done is around between $6 and $10 million. And about the estimate that we have is as Merit was saying, between 1 and 2 years, mainly 2 years.
Yeah, I would have guessed 10 without having any quotes or anything, but because it's probably going to be a gene therapy type delivery, I would think. Though you never know, maybe there'll be another way. Yeah.
So there's always the question is it best to use tools that were already used, for example, viruses that are in clinical use today, and not invent new modalities for bringing the drug to the nervous system, which will take more, a longer time. So there's these two options. Maybe they need to be developed in parallel, but it's all about money, the funding. Our desire is to treat patients as fast as we can. That's the idea.
Every breakthrough has a bottleneck. What's the hardest problem standing between this science and real-world impact? Is it delivery, durability, safety, or something else entirely?
So we just talked about money. Well, obviously this is a limitation, but it's not a bottleneck, I would say. And the bottleneck that many of us researchers experience is is an inability to replicate results that we saw in mouse models in human patients. So drugs that were very promising based on trials in mice, now we know that it failed in humans many times. From our point of view, now that we could use human-derived cells and we have clinical evidence of the mechanism that we found, we think that this bottleneck is much less narrow than it used to be in the past for other trials. Obviously, we need to get more evidence from humans that this treatment is relevant and to understand exactly what is the group of patients that it would be most efficacious in. Amir, if you want to add over this, so.
I think as you said, Tim, it's delivery. Delivery is a big problem. Even if you have a good molecule that is really effective, if you cannot get it through the nervous system and into the cells, into the neurons, that's a big problem. So the delivery is key. Fortunately, there's the tools today already developed, so we plan to use things that are not new. But that's a key problem in the local production of this microRNA. But again, that also there's tools that are already in use, but we have to prove that it does use and effective for ALS. So this is the real world stuff. Yeah.
We've seen for the first time recently that ALS can be treated in certain genetic forms. How does your work fit into that broader shift from untreatable to treatable but complex?
You're right. So first of all, indeed, genetic treatments are amazing. Like treating a genetic or a specific gene and correcting it is really state of the art. And as Mary showed us or told us, this is really helpful for patients with specific mutations. However, still most of the ALS patients don't have any known genetic background. They are sporadic or idiopathic. So there's still a big group of patients that could really benefit from such a treatment. Also, I think that combining the treatment with these microRNAs in patients that already received the genetic treatment would really help to facilitate the regrowth and regeneration of the nerves and the recovery back to function.
Maybe used as a dual mechanism. Absolutely. It tackles different parts of the system. So it can be really beneficial. However, this microRNA treatment is not selective. It's not effective only in sporadic or only in a hereditary disease. It should be worthwhile for both.
Does this mechanism suggest a therapy for a narrow subset of ALS, or does it point towards something that could matter for a much wider group of patients?
So it does matter for a wide group of patients and not selectively to any particular group.
How much does timing matter? Is there a point of no return in ALS, or does your work suggest there's more resilience left in the system than we once believed?
Right, so this is debated. Generally, it's probably the early treatment is best because the ability to regenerate is better when there's not a lot of damage to the nerves. However, as Mary pointed out a few minutes ago, or earlier, then we can see regeneration of nerves even in severely affected patients. So that still needs to be looked at, and probably even when there's severe weakness, there is some ability for regeneration. And particularly, this treatment showed that it pushes regeneration really nicely, not only stopping the mechanism from moving forward, but also regeneration. So that needs to be addressed, hopefully, when we reach a clinical study, maybe also in animals beforehand.
You work with data every day, but ALS is a human disease. When you think about someone diagnosed last month, what do you want them to understand about what this discovery does and does not mean for them right now?
First of all, we're in a new era where now we are understanding more about the mechanism of this disease. I think we don't need to, use medicines that are just taken from the shelf. Let's try a bunch of different vitamins and medicines that are helpful for other disease, antibiotics and stuff, which don't address the mechanism. Now that we know the mechanism and that we know that molecules can specifically tackle development of this mechanism, then it would be better to address this. So I think that brings hope to patients that are diagnosed now, and hopefully within a few years, we'll have an effective treatment that targets this mechanism.
I would also add two more points from my perspective. First of all, I got to meet a lot of patients here in Israel, and the patient population is really embracing in Israel, and they have a lot of support. So many patients that I spoke with don't think about themselves. They think about the ALS patient populations currently and also in the future. If I'm not getting the appropriate treatment, I want the patients that will follow, that will be diagnosed in 1 or 2 years or 4 years. I want them to to get the most effective treatment, and they work a lot as a community to promote research and promote science and new treatments. The second thing is that I think that there are already in the pipeline some treatments that have been identified prior to our finding that also can try to target basic mechanisms of the disease, and they I think are more advanced than where we are standing now. So they might be a good option for patients in clinical trials that might open soon, and it's not like there's no hope at the current state. We are moving as fast as possible to develop our treatment because we believe in it, but there are also other treatments that seem to be promising, and I hope that eventually patients would benefit from those.
Yeah, Mira touched on this earlier, but when my dad was diagnosed in 2016, but we really didn't do anything until 2017. And man, it's crazy how different it is, the landscape. ALS there was one drug that could slow it by 10%, and then there was a big thing that maybe they thought they could slow it by 40%, and maybe together they would stack. And now we're talking about repairing and regenerating. It's exciting. I say to people, I get a decent amount of calls from people who get diagnosed with ALS, like families, and they call to ask us for advice. And I say to people, like, you never want to get diagnosed with ALS, but the best time to do it is right now. Like there is some stuff actually happening. 20 years ago, there's really nothing you could do. So it's terrible to say, but it's just the truth.
There's a fine line between hope and hype. As scientists, how do you stay grounded while working on something that naturally raises expectations?
You're right. There's definitely hope and hype. I think that every finding by itself, every scientific research that is being published creates a hype. But all of them, summing all these up together, as we just talked about the advancement in the research in recent years, when you sum all of these findings together, each one is a hype, but together they create hope, I think. So I think also our research is partially a hype, but also a hope, because we really combined several evidences that were identified before, but what we put them out together, and now we also suggest a way to treat it.
There's lots of different details which we don't know, but we're hoping we have enough to push forward for treatment. We're trying to figure out the other things as well in the meantime, but we're never going to know everything. So we have enough to move forward. That's the idea.
Looking ahead 5 years, what would success actually look like for you? Not in papers published, but in how patients might experience this disease differently.
Yeah, in five, so we were talking about maybe in 2 years we can reach a phase 1 clinical trial. In 5 years, I'm really, really hoping that we'll be in a phase 3 or even using this drug to stopping or treating, curing ALS. That's my hope. In 5 years, really, really hope to get there.
I was thinking it would be definitely a proud moment, but I'm not looking for any pride in my own specific research. I think, again, summing all the achievements that were made in the ALS field in the recent years, it would be extraordinary to see some of this translating into results in patients. Obviously, if our clinical trial would work in patients, I would be the proudest father in the world, knowing that I've been able to help so many people. And hopefully I could also reply back to all the emails that I've been getting, and as soon as also Amir has been getting. Currently we replied that it would take time and we still need to develop, and hopefully in the future my replies would be different. You need to take this works. But it's yeah.
If I'm going to take a more negative approach to this for a second. If in 5 years from now it didn't work, would it be something with the problem with the science? Would it be a problem with fundraising? Would it be a problem with safety? What would you guess the problem was? If I told you in 5 years ago it's not in people, what would you guess?
I don't suspect that there would be any safety issues, but I need to be cautious. I think it's more about the delivery, getting it to the right place in the right amount and the right timing, and we would keep working on that, trying to improve. A drug has a very long life, so you try once, you understand and learn the mistake or the problems, and then you improve your drug, unless it's a complete failure. But we don't think that this is the case in our treatment.
There's a lot of work to be done. Okay, and in science, whenever you try to take a shortcut, you find yourself wasting time. So this really needs to be developed cautiously, systematically, like we learned and like we do usually, and using different doses, making sure there's no toxicity and the delivery is right and the distribution is right. And this takes time and lots of money. And if we don't get there in 5 years, I think it would be because we weren't able to invest in doing this properly from whatever reason, money, time, resources, etc.
And what does this look like now on your guys' side? Is this now like its own company, its own venture, or is this just research and somebody else will kind of take it and run with it, or what? I guess what now? Like, is this like does this thing have its own legs? Like Ariel, you said feel like a proud father. Is this like a company at this point, or what is it?
So I think it's more of a venture, and Amir can elaborate on this.
I think it's in what's called in the value of death. which means that it's not academic anymore, but companies don't want this yet. So we're still trying to raise the money needed to get through the next stage, which is delivery, showing that if we deliver it in the right way with the right tools, then it's effective. And after that, hopefully it will be successful, and then company or some big pharma will want to invest in it, and then it will be pushed forward. But currently, there's a lot of fundraising to do the next step, preclinical step.
What's one quiet truth about ALS research today that doesn't make headlines but gives you genuine confidence that the field is finally moving forward?
It's not that quiet, but I'm thinking of we've talked about it before about the human-based models that shifted the research. Now we know how to generate human-derived motor neurons and human-derived skeletal muscles, and actually regenerate the entire motor unit in a dish. And we can test multiple drugs and multiple treatments at the same time in a human relevant model. There have been many failures in trying to translate findings that were just relevant in mice, but now we can shorten this by working directly on proper human models. That's what I think it has been, maybe the quiet truth, but it really advanced the research in the recent years, and it's becoming more and more common in research labs, and actually a standard of research in ALS.
I think as a clinician, there's debates about, is ALS one disease which affects both the central nervous system and the peripheral nervous system, or these are two diseases which sort of eventually become one, but they start as two different disorders. That's hard to tackle, and it's hard to answer. Our system, our lab, and our tools are focused on the peripheral nervous system because that's the nature of our lab and my clinical field, which is biopsies and EMG and that sort of stuff. It's harder to know what's going on in the central nervous system from this perspective. So we still don't know about that, but we will learn.
Before we close, take a minute and brag about your families. Who are the people who share you with this work? And what do you hope they understand about why you chose to spend your life chasing answers that may take years to arrive?
Even in this moment, outside the door that you can't see, my wife is trying to put to sleep our 3 children, and she's been doing it a lot during the work in, I guess, the 8 years since our first daughter was born. There have been many occasions where I was locked up in the lab or in the microscope trying to find answers for all sorts of evidence that we identified. And first of all, a lot of my achievements are, I owe a great thanks to her. And it's for my children. I think I see it in their eyes. They understand that what we do is important, and it has a greater impact than just doing any other regular daily job. So they also spent a lot of time in the lab together with me when we didn't have any other solution. So they got to know the lab life and the both the more happier moments and the moments that are more difficult when an experiment fails. But resilience, I think it's a good lesson to be learned in their case, and I try to teach them how to deal with failures and how to keep on going, and that nothing is unachievable. That if they want to chase something, they can do it and they can achieve it. That's me.
My kids are older, so I can tell you that earlier when I would criticize them for being on the internet all the time, using their staring their tablets all the time. So whenever I would do that, they would say, Dad, don't you have a biopsy to look at, or go see some biopsies at the lab? So they were, in a way, pushing forward, the my education all the time for years. They were supporting me studying and researching and going to the lab and going to the hospital. My kids and my wife, and very supportive. Absolutely. I owe everything to them.
Hey, one other thing. How about AI? Has that made a big impact on what you're doing?
So I think it hasn't really stroke yet in the daily life in the lab, at least in our type of research. So it's developing. I'm seeing it also from a newer perspective in the pharma companies. AI is getting stronger and getting more incorporated into science, into identifying in new targets, into analyzing the data, and really joining points that we could, as people, as scientists, cannot join together.
In the clinical world, it's great. Quick answers for, in the real world where you need a quick answer. And also the load in the emergency room and in departments is so high. Now we're starting to use AI for triage, of patients. And I think it sounds really bad, but once you experience it, then it's gonna really make a big change and make things actually work for the better.
Now on to our final word segment, where we ask our guests a few rapid fire questions. What was the happiest moment in your life?
When my daughter's my daughter was born. I think it was a life-changing event.
It's actually, same answer. When my kids were born, that was the happiest moments.
What is the biggest adversity you've faced?
I think, my father passed away when I was relatively young and not really knew a lot of things about life. So I had to spend most of my adult life thinking the thoughts instead of hearing my father's advices. And I learned to do that and support my mother and support the entire family.
Death in the family was the hardest.
What are you most excited about?
The unknown. I'm excited about the unknown. I don't know where life will take me. I know where I want to be and where I want to go, but the unknown is definitely exciting.
I think these things that we were just talking about, for many years I focused on the clinical work, and now that there's this science and the translating the science back to the clinic, I think that's the most exciting thing from my point of view.
What advice would you give your younger self?
To make the life-changing decisions faster, although sometimes you can't really control it.
Exercise more. Yeah.
I have good answers. I mean,
Do you have any book recommendations for us or for the listeners?
I don't have a lot of time to read books, so actually I'm listening to books on audiobooks, and it's something that I started quite recently to do in the last year or so. And there is this actually crime book series of Cormoran Strike, wrote by Robert Galbraith. It's actually J.K. Rowling pen name, and it's really, really good.
Also, I unfortunately don't read books now, but my wife wrote a book, so I can recommend that. It's about kids with special needs. I recommend that.
The name of our podcast is Nothing Left Unsaid. Do you have anything you want to say?
I just want to thank the support that we've been getting from patients, from people, from obviously from my family. As I just thanked before, um, for supporting science. You know, the funding agencies that supported my research, my education, and brought us to this point.
Also, thank you for the patients and the families. This is really lots of suffering. I see it in the patients' eyes and in the families, and it's hard to. I mean, I see it, and their participation in research, particularly giving a muscle biopsy or blood samples. Really, really thankful for that. It couldn't have happened without them. Without them.
Do you have any recommendations of people you know personally that you think we should try to get on the podcast for future episodes?
Wow, it's a tough one. Um, Maybe Donald Trump. I think he has a lot of answers for many questions. And I think maybe Amir can come up with a different, more realistic option.
Yeah, I don't know. My mentor in this field, his name is Alan Pestronk, and he's from Washington University in St. Louis. He taught me much of what I know. And you know, whenever I see a muscle biopsy, and particularly along this process, I was hearing him in my head, and sort of quotes that he would say. And he's about 80 now, and he can describe, you know, very long way of things that happened with ALS. So many patients follow the developments in this field and taught me. So I recommend him, Alan Pestronk from Washington University.
Ariel, Amir, thank you for taking the time to walk us through this carefully and honestly. One of the things Merit has taught us on this podcast and off is that real progress in ALS doesn't come from breakthroughs alone, but from scientists who are willing to explain what they know and admit what they don't, and stay connected to the people living with the disease. Today felt like that kind of conversation. You help us see how the science is moving forward without pretending the work is finished. We're grateful for your time and for your clarity and for the families who support while this work unfolds at its own pace. Thank you for joining us today. May God bless you, your families, and the work ahead.
Thank you for having us and for giving us the opportunity to explain what we do and the meaning of what we do. It was a great pleasure to be here today.
Yes, thank you very much. Very grateful for this talk. It was a great opportunity.
All right. Thank you.
Thanks, guys. Look forward to the one in the future when we get it in people and it's working, right?
Yeah.
Right, exactly.